Welcome back to Study Tips for IGCSE Biology. Today's topic is related to biotechnology. Without further delay, let's begin. In this chapter, you will find out about features of microorganisms that make them useful in biotechnology, the use of yeast to make biofuel and bread. We use enzyme in biological washing powder, the use of pectinase for making fruit juice, the use of lactase to make lactose-reduced milk, penicillin is used to make penicillin, and some example of genetic engineering. What is biotechnology? Biotechnology involves using living organisms to carry out processes that make substances that we want. Usually, the term is used only when microorganisms are involved or when plants or animals are used to produce something other than food. Example, microorganisms to make various products such as yeast. Yeast is used to make bread and alcohol. Bacteria has been used to make yogurt and cheese. Why we use microorganisms? Biotechnology and genetic engineering often make use of microorganisms such as bacteria and microscopic fungi. There are several reasons for this. First, bacteria and fungi are very small, easy to grow in lab, and do not take up space. In addition, they reproduce very quickly and make a huge range of different chemical substances. Second, bacteria cells are very different from animals and plant cells. The genetic code of bacterial DNA, same as human. As such, we can take a gene from a human cell and place it into bacterial cells, and it will work to produce the human protein. Third, bacterial chromosomes have little loops of DNA called plasmid. Plasmid make easy to transfer from one cell to another and also moving genes from one organism cells to another. Yeast is able to respire anaerobically. When it does so, it produces ethanol and carbon dioxide. Below is actually chemistry equations where glucose produces ethanol plus carbon dioxide during fermentation. This process is called fermentation. Making biofuels. One alternative is to provide sugar, which yeast can then break down to form ethanol. Ethanol, sometimes called bioethanol. Bioethanol can then be used as fuel. Maize is one of the crop that is used in this process. It is first treated with amylase enzymes, which break down starch to glucose in anaerobic respiration. Below is a chemistry equation where you can see amylase enzyme has been broke down starch to glucose. This process is called anaerobic respiration. This process is actually happening during anaerobic respiration. The ethanol produced can then be extracted from the mixture of biodistillation. Alcohol burns well, it does not contain as much energy per liter as fossil fuel. So it's then mixed with gasoline petrol to make fuel used for vehicle engine. Advantage of using biofuel is that it sustains resource and reduces the amount of carbon dioxide in the atmosphere. Carbon dioxide is produced when the biofuel is burned. The plants that we grow to make fuel took carbon dioxide from the air. Then they made the sugars and starch by photosynthesis. Making bread. Bread is made from flour, which is made by grinding the grain seeds of cereal crops. Most bread is made from wheat flour. Flour contains a lot of starch and also protein called gluten. To make dough and flour is made with water and yeast. Amylase enzyme break down some of the starch in the dough to make maltose and glucose, which yeast use anaerobic respiration. Dough produces bubble of carbon dioxide. Gluten makes the dough stretchy, so the carbon dioxide bubbles cause dough to raise. Anaerobic respiration is all broken down when the bread is baked. Baking also kills the yeast. Making use of enzyme in biological washing powder. Many different enzymes are used in industry. Most of them are obtained from microorganisms. The microorganisms are grown inside large vessels called fermenters. Inside the fermenters, the microorganisms are provided with everything, including oxygen, nutrient, suitable pH, and temperature. The microorganisms make enzymes and release them into the liquid in which they are growing. The liquid can then be collected from the fermenter and purified before use. Biological washing powder contain enzyme as well as detergent. The detergents help greasy dirt to mix with water so it can wash away. The enzyme help to break down other kinds of substances which can stain clothes. They are good at removing dirt which contain color substances from animals or plants like blood or egg stains. Some of the enzymes are protease which catalyze the breakdown of protein molecules. This helps with the removal of stain caused by the proteins such as blood stain. Blood contains the red protein hemoglobin. The protease in biological washing powder break the hemoglobin molecules into smaller molecules which are not colored and which dissolve easily in water and can be washed away. Some enzymes are lipase, which catalyze the breakdown of fat to fatty acid and glycerol. This is good for removing greasy stain. To prevent this enzyme from digesting proteins and fat in the skin of people handling them, the enzymes are packed into microscopic capsules. The capsules break open when the washing powder is mixed with water. Pectinase. Fruit juice are extracted using enzyme called pectinase. Pectin is a substance which helps to stick plants together, plant cells together. Fruits such as apple, orange contain a lot of pectin. If pectin is broken down, it can be much easier to squeeze juice from the fruit. Pectinase is widely used commercially both in the extraction of juice and making the juice clear rather than cloudy. Lactase. Lactase is an enzyme that breaks down lactose, the sugar in milk. Lactose, in the process of lactase, to produce glucose plus galactose. All human babies produce lactase in their digestive system. This helps babies to digest the lactose in the milk that is their only source of food for the first few months of their life. Milk can be treated with lactase to break down the lactose so that people who don't make lactase themselves can drink milk or eat products made from lactose-free milk. Another reason for treating milk with lactase is to produce glucose and galactose, which can be used for making sweets. Penicillin is used to make penicillin. Antibiotics are substances which kill bacteria without harming human cells. We take antibiotics to help to cure bacterial infections. Penicillin is made by growing the fungus Penicillium. Penicillium is grown in a culture medium containing carbohydrate and amino acids. The fungus just grows. The stage takes about 15 to 24 hours. After that, it begins to secrete penicillin. The rate at which it produces penicillin partly depends on how much penicillin is made. The small amount of sugar has to be fed into the fermenter all the times that the fungus is producing penicillin. The culture is kept growing until it is decided that the rate of penicillin production has slowed down so much, approximately about a week. Genetic engineering. Genetic engineering, also called genetic modification, is a process that uses lab-based technology to alter the DNA makeups of an organism. This may involve changing a single base pair A, T, C, and G, deleting a region of DNA, or adding a new segment of DNA. We have seen that gene is a length of DNA that codes for the production of particular protein in cell. We are now able to take genes from one organism and put them into another organism. This is called genetic engineering. Genetic engineering was first carried out in 1970. Since then, many process has been uses this process, such as insulin. In the process of insulin is now produced by bacteria. Insulin is needed by people type 1 diabetes. The human insulin gene was inserted into the bacteria and they are now grown in huge vats. The process begins with the extraction of the gene for insulin from human cells. This is done using enzymes called restriction enzymes. This enzyme cut DNA molecule at a particular point, give short length of unpaired base at either end of the cut DNA called sticky end. The particular length of DNA that is required, the insulin gene is identified and extracted from all other DNA. One way of getting DNA into bacteria is to use a plasmid. Plasmid is a ring of DNA found in bacteria. First, the ring of DNA in the plasmid is cut using the same restriction enzyme that we use for cutting the human DNA, which will leave sticky ends that are complementary to the one of the human DNA. The human insulin gene and the cut plasmid are now mixed together. The sticky end and pair bases on the insulin gene pair up with sticky end on the plasmid. Another enzyme called DNA ligase link the two strands firmly together. Now there are plasmids that contain the human insulin gene. They are called recombinant plasmid because they contain a combination of bacteria and human DNA. Next, these genetically modified plasmids are added to a culture bacteria. Some of the bacteria take up the plasmid into their cell. These bacteria are put into fermenters where they reproduce asexually to form large population. Second, genetic engineering is used in the process of crop plants. Crop plants has been genetically modified to be resistant to herbicides or insect pests. Example soya plants has been genetically modified so that they are not harmed when herbicide called glyphosate is sprayed onto them. Number 3, genetic engineering is being used in the process of making rice. Rice has been genetically modified to produce much more vitamin A than normal rice. Lack of vitamin A can cause blindness and cause a big problem to some part of the world, particularly where their diet mainly consists of white rice. Growing and eating golden rice rather than ordinary rice could be a big help in solving this problem. Nevertheless, there are some concerns about the use of genetically modified crops, and it is important that these are carefully considered. They say that it would be better to solve the real cause of this problem, which is people do not have enough money or enough food in some part of the world. At least with the golden rice, may be able to help some people now, whereas trying to get rid of poverty is not as easily done. With that being said, I would like to end my presentation, and I hope that you are enjoy studying, and please subscribe to this channel. Thank you very much.